All right, everybody. My name is Clay Tucker. I actually just got my PhD at Louisiana State University. I'm transitioning into a postdoc right now. But today I'm going to kind of continue some of the research that Savannah just talked about from the North American Dendroecological Field Week last year in Wyoming. One of the big advantages to native, as we call it, is that you can come at a problem with multiple avenues. Savannah just talked about quantitative wood anatomy and whitebark pine mortality. And I'm going to talk about the same stand of trees essentially, but from a different perspective, the isotopic perspective. I do want to acknowledge the two co-authors and group leaders at Native. On the left, Justin Rose, and on the right, Adam Chank, who you heard from earlier with Pyro work. And I also want to thank Chris Gentry and Adam for organizing this. This has been a great experience so far. But I want to start with why we choose isotopes. When we do tree ring width research, we always leave with a lot of questions. And of course, we're going to end with questions today. But we always end up with a lot of questions like, well, was it rainfall, or was it temperature, or moisture stress, or was it fire? Why did a tree grow in this particular way in this particular year? And the reason we go a little further, or we spend the money essentially to do isotope dendrochronology, is because it gets us further into reasons why something might have happened. So just a quick overview on what an isotope is. Every element, or most elements, have multiple isotopes naturally in nature. And we've also, for this example here, we've also produced a radioactive one for the hydrogen bomb known as tritium. But essentially an isotope is a molecule with an extra neutron in its nucleus. And so we have heavy isotopes and light isotopes. And I may use these words enriched and depleted as we go forward, but just know that these things occur naturally. And we have this ratio that changes based on environmental factors and can be really helpful in getting deeper into the questions of dendrochronology. For the most part, methods are all traditional except for three big exceptions I'll say today. The first one is represented here by the increment borer. If you can see at the top left image, this hand is either really tiny or the increment borer is really large. This is a 12 millimeter increment borer, often known as an isotope increment borer because the width of the core is much larger. And the reason we do that is because we're actually going to sample each individual ring. And this is on a micromill. This was for some of the samples that we did last year at Native. And we basically just drill or mill each ring individually. Sometimes we separate into early wood and late wood. And then we'll do some extraction methods, some chemical extraction methods to do just cellulose. There is a video there, but I'm not going to run it. So we extract cellulose. Cellulose and lignin are the two big parts of wood, and they use different nutrients at different times. So we get it all the way down to alpha cellulose, which is a pretty good marker for the kind of carbon and oxygen and hydrogen that the tree is uptaking at the time of wood production. So that's two of our reasonings behind why we do isotopes. One, because, or the difference in methods of isotopes. One, we use a different increment borer. Two, we do some chemical methods. And three, of course, we run it through a really expensive machine to get out isotopes. And what carbon isotopes, we're going to talk about carbon isotopes today. What carbon isotopes reflect is something called stomatal conductance. And so the leaf on a tree has these small openings in it called stomata, and they open for gas exchange. And when they're open, the tree can release water and oxygen. When they're closed, they can't do that so quickly, at least. And so the tree will have different concentrations of CO2 in it. The tree truly wants to take in whatever carbon dioxide it can at all times, but it's going to most easily process the lightest of those isotopes. So it's most easily going to process carbon-12, which is represented here in the normal font, but it will also uptake carbon-13 when the stomata are open. it just won't process them. So when the tree is moisture stressed, these stomata close so that it doesn't lose water, and the tree is forced to use its carbon-13. So you get a more enriched field during moisture stress times. So again, the carbon isotopes that we talk about today reflects stomatal conductance, and then from now on, I'm going to use the phrase moisture stress a lot. So two major questions we have, very similar to what Savannah talked about previously. What's causing the mortality here? One, do the isotopes follow moisture stress as they have in other studies? And if so, which climatic variables show the strongest correlations? And then if we can, just like Savannah aimed, is it the moisture stress, or is it some other factor causing mortality to these whitebark pines in northwestern United States? So here is, I'm going to keep it pretty simple for today's graphics. Here is a time series of 31 years, I think, of data, carbon isotope data. This is what we call big delta, so discrimination, from 1986 to 2017, I believe. And the way I want you to think of this line is, as this line increases, or as this line decreases, so this low point here in 1996, you have a high moisture stress. So the way I really like to think about it is, as this line increases, we have happy trees, and as this line decreases, we have unhappy trees. So a peak in moisture stress here in 1996, and sort of a gradual recovery, though another drop here in what I believe is 2007. So from the trees that Savannah talked about earlier, we know that there was a large death of trees in the late 2000s, let's say. And so we can kind of see that this marks a period of moisture stress in our trees based on the carbon isotope data. The other thing that's really interesting to note from the carbon isotope perspective is that it may provide us some kind of point at which the bugs introduce themselves to the tree, or where the trees were heavily stressed and the bugs could have come in. But the trees, you know, the moisture stress throughout the late 90s and early 2000s wasn't too bad. But again, in this late 2000s, we had some more moisture stress. So there may have been an infiltration here in the late 90s, and then a die-off here later when moisture stress increased again. So just some simple correlations. We picked out two typical ones: temperature and precipitation. Temperature had a better signal, 23% of variance. And essentially, during hot, dry summers, trees are less picky for the carbon that they process. So the really big picture here is that moisture stress is a big driver or a big correlation to what's happening to these trees before they die. We also did some daily analysis. I can talk to this a bit in the questions. It would take a long time to explain, but essentially you take in some daily data to correlate exactly when these trees are producing wood. Since these trees aren't growing, a lot of the times the trees I work with in the South are growing for 9 or 10 months out of the year. These trees might grow for only a few weeks. So we did do some daily analysis. For temperature, we're seeing a signal later in the year, and for precipitation, a signal a little earlier in the year. So just some overarching conclusions. Tree ring delta 13C, this carbon isotope work that we did does reflect moisture stress. High summer temperatures with low precipitation, hot, dry summers cause those trees to close their stomata, thus processing more of that heavier isotope. And as with the normal chicken or egg hypothesis, we can't really confirm anything. However, moisture stress does explain 23% of the variance prior to all of that whitebark pine death. So we may not be able to say that it was just the bugs or just moisture stress that caused mortality, but we can say that moisture stress probably enhanced the ability for the bugs to come in, the mountain pine beetle, I believe, and may have contributed to this explosion of mortality. That's all the time I have for today. I do have my information there, and as usual in most of these presentations, I always like to give a tip of the hat to Jim Spear and the whole Native community. I don't have tree ring science at the University of Louisiana State University, where I'm from, and where I did where I did all of my PhD. So that was a really helpful part of my studies, my tree ring studies through time. So with that, I'd be glad to take anybody's questions. I see some here.
All right, so we got one question that's come in so far. How do you know the best part of the tree to sample for this work?
That's a good question. We still follow all dendro principles until isotope preparation. So we still cross date, which means we still want, low in the tree. We still want as many years as possible. So we go low in the tree. We still core to pith if we can. So it's the same traditional methods until you get to isotopic preparation.
So there's another question. What was your sampling design? Number of trees, annual years pooled, and are these consistent with ring widths?
That's a good question too. One of the downfalls to dendro isotopic work is that it's expensive. So you can probably start a whole entire dendro lab for the cost of these samples. I believe this is from 6 cores, 1 core per tree. Because it's an experimental study, exploratory study, excuse me, we just use 1 core per tree, but they do all cross-date with each other, and furthermore, they cross-date with some of Savannah's trees and some of the trees that were used in a dendroclimatic reconstruction for this area too. So a low end for ring width studies, but pretty traditional and typical for isotopic studies.
Question for you, Clay. How do you differentiate low 13C related to hot summer compared to bug presence? Does atmospheric variance play into 13C counts?
Oh boy, I see that's from David. That's a really good question as well. I always say that. You can't really differentiate between hot summer compared to bug presence. I can't say it's one or the other. But what I can say is that the 13C in our study did correlate to, and the variance was explained by temperature and precipitation. So moisture stress is a likely driver in growth, or at least in the isotopic composition of these trees. And yes, atmospheric variance plays into 13C counts. We take that into account through mathematical formulas. Things like we've obviously increased CO2 concentrations in the atmosphere through fossil fuel burning. And that's taken into account through some mathematical processes post receiving the data, the isotopic data, raw isotopic data.
All right, and probably the last question for Clay. Can bugs induce some sort of feedback loop into the big delta?
I'm not sure what the big delta is. Oh, the big delta. Yes. Got it. I'm not from that area, so the jargon is just coming back. But I guess it could. There could be some kind of feedback loop to where they induce stress or increase stress, but I think it's following atmospheric loops a lot more closely than bug-related growth changes. I don't know. That one will have to be one I think about.